We are on ESMO today, and we will have the pleasure to present the final analysis of GP2013 biosimilar data. This is the largest ever biosimilar trial with over 630 patients on board, randomized between RCVP and GP2013 CVP in untreated follicular lymphoma patients. In the largest ever Rituximab biosimilar trial, we demonstrated the similar efficacy of originator Rituximab and GP2013 biosimilar. It is in fact the end of the process, the end of the long story. The biosimilar development is based on the preclinical studies and a lab assessment where we prove that the molecule is actually identical to the originator. We prove the biosimilarity by all accessible tests we have, and based on that, we can confirm the pharmacokinetics, dynamics, and efficacy in the clinical trials. This randomized third phase, a double-blind controlled trial. The results we are presenting today is the end stage of the whole process, where we can prove the biosimilarity of GP2013 particle. The biosimilar development pathway is based on preclinical studies where, in a very thorough way, we assess the particle by all accessible methods. Later, we have the pharmacokinetics and dynamics study, which were performed in case of Rituximab biosimilar in rheumatoid arthritis. This is the population where we give the Rituximab injection just twice, followed by 6 months of doing nothing, so an ideal setting for this purpose. Follicular lymphoma was chosen to prove the efficacy in oncology patients, as it is a relatively unique disease with a low lymphocyte variability. With 634 patients on board, we were able to prove that the overall response rate is identical to originator Rituximab. This was a primary target of the trial, and this done with 95% confidence interval. We have completed the process in which we proved the total biosimilarity of the particle. Therefore, we are now allowed to use it not only in rheumatoid arthritis and follicular lymphoma patients, but in all indications that Rituximab is registered to. This is called an extrapolation, and it's allowed if a totality of biosimilarity is proven based on preclinical trials followed by what we completed.